Hello, everyone. I am Laura Mondano, and today I'll be talking about sexual behaviors and reproductive endocrinology of subadult female woolly monkeys before dispersal. So before I begin, in respect to other Spanish-speaking scholars, I have included a brief translated summary. En la parte superior podrán encontrar los puntos más importantes de cada diapositiva porque hay que recordar que la ciencia también se habla en español. Okay, so sex-biased dispersal is one mechanism that may help animals avoid inbreeding with kin. In many male philopatric species, where females are the ones that disperse from their natal groups, female reproductive maturity does not occur until after females have dispersed into new groups where they can mate with unrelated males. In adeline primates, for example, they're characterized by having female-biased dispersal, and in both spider monkeys and muriquis, subadult females tend to disperse before they begin to display sexual behaviors or show any evidence of ovarian cycling. In woolly monkeys, however, subadult females have been observed displaying sexual interest towards co-resident males and copulating with adult, subadult, and juvenile males prior to dispersing outside their natal groups. However, it is still unclear whether females start ovulating before or after emigration, and therefore what we wanted to do was to investigate the relationship between ovarian hormone patterns and sexual behaviors in subadult females prior to them dispersing from their natal groups. To do this, we studied a population of wild lowland woolly monkeys at the Tiputini Biodiversity Station, a field site located in the Amazon rainforest in Ecuador. This is part of a long-term study on 4 social groups of woolly monkeys that we've been following since 2014. And during the 2016 mating season, we followed Gabby, a subadult female natal to group G, who we were able to collect data for 4 months prior to her dispersal. And later in 2017, we followed Cuenca, a subadult female from group C, who dispersed about a month and a half after we started following her. We recorded sexual behaviors in the context of focal animal sampling, including female-to-male solicitations, copulations, and male inspections to the female's anogenital regions or their urine. To create individual hormone profiles, what we did is that we extracted steroid hormones in the field by mixing fecal samples with an alcohol solution, and then we stored hormones in a solid phase extraction cartridge, which we then brought to the US, where I analyzed hormone concentrations by conducting enzyme immunoassays on fecal progesterone and estrogen metabolites. And then I estimated ovulation based on sustained increases of PDG, followed by an increase in E1G. And for anyone that has conducted these type of assays, they know how important a good playlist is to get you through this very monotonous work. Okay, so what we found is that Gabby's hormone profiles indicated that the first confident estimate of ovulation was just 4 days prior to her disappearance, which is presumably when she dispersed from her natal group, which is interesting because it's around the same time when we first saw her copulating and displaying sexual behaviors, such as soliciting males via perceptive behaviors like teeth chattering. And also it was the first time that we saw males inspecting her anogenital region. Unlike Gabby, however, we saw that Cuenca did not show, she did show an increase in ovarian hormones right before her dispersal, but this rise did not meet the criteria to assess it as an estimated ovulation. And also, unlike Gabby, Cuenca was never observed displaying sexual behaviors before emigrating her natal group. So, although our sample size is small, what we see is that the increases in ovarian hormones in these 2 subadult females prior to them emigrating from their natal groups may be indicative of the onset of puberty prior to or close to their dispersal to seek new social groups and potential mates. Additionally, genetic and demographic evidence suggests that female woolies may conceive soon after dispersing from their natal groups. This is the case of a different female who was initially followed as a subadult in group P in October of 2015. And then in 2016, she was observed ranging with group G. In September of 2016, actually, this female gave birth to an infant who was later confirmed to have been sired by a resident male in group G. And considering that in this population, the gestation period is an average 7.1 months, this female probably conceived with the male in group G sometime around February of 2016, which is just a few months after she was last seen ranging with her natal group P. Therefore, it is likely that female woolly monkeys reach sexual maturity and become conceptive soon after dispersing from their natal groups, which is different to what we see in other adelines, including the sister taxa, the muriquis, where subadult females tend to disperse before they begin to display sexual behaviors or show evidence of ovarian cycling. These differences between woolly monkeys compared to other adelines may be explained by the fact that home ranges in woolly monkeys overlap substantially, and therefore intergroup encounters are more common than in the related muriquis or the spider monkeys. Also, it's important to keep in mind that 60% of these intergroup encounters are not marked by aggressive interactions, and instead, they are considered to be neutral or even affiliative, where individuals from different groups range and forage together. Additionally, it's not uncommon to observe males scent marking during these intergroup encounters, like this male on the left. So it is possible that subadult females are being exposed to chemical signals from extragroup males, which may actually help trigger the onset of puberty before females emigrate from their natal groups. Similar observations have been recorded in wild geladas, where females were found to be 3 times more likely to mature after exposed to a non-resident male in the context of a takeover. And therefore, what we see is that social conditions, such as the exposure to novel males, may trigger the process of sexual maturation in subadult females. So in conclusion, what we see is that it is likely that a high degree of home range overlap and frequent encounters between animals from neighboring groups may expose predispersal subadult female woolly monkeys to chemical signals from novel extragroup males, which may induce the onset of puberty before females emigrate. I first want to thank the Waorani and Quechua indigenous people who have resisted for many years against forest destruction and have been the protectors of the forest where we have had the privilege of conducting our studies with woolly monkeys. I also want to thank all the staff and field assistants at Tiputini, as well as the funding agencies that made this project possible. And finally, thank you very much for watching my talk, and please contact me if you have any questions. Thank you very much.